Genetics is the study of the inheritance of traits from the parent to the offspring. Genes control the type and timing of the production of proteins. And those proteins then determine the characteristics or the structure and also the metabolic processes of an organism. The genetic makeup of an organism can be called the genotype. And how that's expressed in the structure and the metabolic processes is called the phenotype. Much of the early work on genetics was conducted by an Augustinian monk named Gregor Mendel. And he made very, very detailed and careful observations about pea plants. He was able to identify a number of traits, and he was able to breed these pea plants so that they were pure breeding for these particular traits. So pure breeding, what I'm going to do is I'm going to start to write a list of definitions here, and in my summary I'm going to write what these definitions mean. But pure breeding means that each offspring has those particular traits. So, for example, one of the things that he looked at was the type of seed, whether the seed was smooth, whether it was a smooth seed, or whether the seed was wrinkled. So, it's a wrinkled seed. So he bred these plants until they were pure breeding for either being a smooth seed plant or a wrinkled seed plant. And then he crossed them. So he artificially pollinated them, so he had he could very much control how they were being pollinated. And so he crossed the smooth seed and the wrinkled seed, and he made an observation about the offspring. So that's called a cross. And when he did that, in the first generation of offspring, the F1 generation, sometimes we call that the filial generation, or easier just to say the first generation. So he crossed his smooth seeds and his wrinkled seeds. Every one of the offspring of this cross in the first generation was smooth seed. So only smooth seed offspring occurred in the first generation. So Mendel thought that what that meant was that being the smooth seed must be the dominant trait. So he thought it was the dominant trait. And so we can express this and explain it in a Punnett square. So again, more terminology we need to put up here. A Punnett square. But a Punnett square is a way that we're able to describe the cross between these two pure breeding plants. Now, you'll recall that an organism has the chromosomes are paired, and on each of these homologous chromosomes they have the same gene, but they may be different types of that gene or different alleles of that gene. So a Punnett square is a way of predicting the outcome of a cross between two organisms. So each of these squares represents the possible genotype for a particular trait when there's a cross between two parents. And so we have one parent up here, we have one parent down here, and in this position here we would say write one of the alleles, and here we will write the other one of the alleles. And for the other parent, one allele and one allele. So remember, when you have sexual reproduction, you have one chromosome from one parent and one chromosome from the other. So each contributes an allele. So we have an allele from the mother, an allele from the father, and we combine that, we get the genotype for that particular offspring. In this particular case, if we've got a pure breeding plant, it must have the same allele. So the convention we have here is for the dominant trait, we use a single letter to describe something about that dominant trait. For example, smooth seed, we're going to call that capital S. So the dominant is the capital, and we're going to put the smooth seeded plant up here at the top. So capital S, capital S. Because it's pure breeding, it must be homozygous. So homozygous means that both alleles are the same. And so for our wrinkled, we're going to put the alleles for our wrinkled plant here, and we're actually going to call that little s and little s. So we don't call it a W to describe the allele. We're suggesting that the wrinkled one, because the wrinkled offspring didn't feature in the first generation, Mendel says that must have been a recessive characteristic. So we express that as a little s. So when we cross these, we take, there's four possible outcomes in terms of what the alleles will be in the offspring. So we will have the opportunity for a capital S and a little s, capital S and a little s, capital S and a small s, capital S and a small s. So all four of the offspring, or the possibilities for the offspring, are a capital S and a small s. This is what we call a hybrid, and we call it heterozygous. And heterozygous means that we have two different alleles. Now, when we have the dominant allele, this capital S, the one for smooth, the smooth seed, whenever that's present, that's what's going to be expressed in the phenotype. So that means all offspring are going to be smooth seed. Okay, 100% of them are going to be smooth seed in the first generation. But let's see what happens in the second generation. We'll call that F2. This is the second generation. And in this particular generation, we're going to cross the offspring. So we're going to cross the offspring here in the second generation. So this is F2. So we're going to do our Punnett Square again. Alright, so this time we have to put up our alleles. We have the capital S and the small s. Capital S and the small s. Alright, so how did we get this? These are our offspring from the first generation, and now we're crossing those. So we've got capital S and is one of the alleles, and the small s is the other allele. So now we need to look at what our possibilities are. Here's one, and it's capital S, capital S. One here is capital S, small s. Here we've got capital S and small s. And finally we've got small s, small s. So the genotypes, in this case, in the second generation, we have one capital S, capital S. So one homozygous dominant. We have two capital S, small s. So two heterozygous. And finally, we have got one small s, small s. So 1 homozygous recessive. So what are our plants actually going to look like? These 3 have a dominant allele present. So we're going to have 3 smooth seed, but this one here has got 2 recessive alleles. It's homozygous recessive. It doesn't have a dominant allele present, so therefore it is going to be wrinkled. So the ratio we have here is 3 is to 1. So this is what we call the monohybrid ratio. So it's a ratio of crossing 2 hybrids, but this mono here means we're only looking at 1 trait. So they're hybrids, they're both heterozygous. It's a crossing of heterozygotes, but only looking at 1 trait, and the ratio we get is 3 is to 1. So 3 dominant phenotype to 1, so 3 of the dominant phenotype and 1 of the recessive phenotype.